Hey guys, welcome to Psychology from Project 45. Today we're going to be focusing on neurotransmission from the biological approaches to psychology. Okay, so what is neurotransmission? So neurotransmission is basically the act of passing information around our brains, like between our neurons, so that we can perform basic actions like thinking, talking, just breathing. Just everything in your body is practically controlled by these neurotransmitters, okay? Okay, so neurotransmission relies on neurotransmitters, okay? So think of neurotransmission as the action and neurotransmitters as the one doing the action. So neurotransmitters are electrochemical signals used in transmission of information between neurons. So there's these little tiny little particles of information. They can be they're very small, okay? They could be ions, they could be little hormones. Hormones are different, but you can think of them as hormones, but for the neurons. Okay, let's move on. So neurotransmission relies on one important feature or one important thing that happens within the neuron, which is called the action potential. Okay, you might learn this in biology if you do take it in IB, but this is action potential. You don't need to know too much of it. You just need to know that it involves the influx and outflux of both sodium and potassium ions. So it is basically how the information gets propagated along the axon of the neuron, okay? So this is the signal, the action potential, and this is the myelin sheath I'm drawing on right now, and it's traveling down the axon here. So information goes here over all the way to the terminal. Okay, so that's the action potential for you. So this is basically a little graph. You don't need to know too much again. Just maybe mention it in your paragraph or so. Okay, now let's move on to our little mechanics, okay? So in a synapse, which is this place over here, this little like space between the two neurons, it's where the information, where the the impulse happens, okay? The impulse is basically describing the action of also like just opening the vesicles and letting the neurotransmitters flow through to the postsynaptic neuron. So we have here the presynaptic neuron, so pre, and then post. So it means before, after. So before neuron, after neuron. So presynaptic and postsynaptic. It's quite nice to use some more scientific terminology in your paragraphs. So yeah, please do include presynaptic. I'll write it here. Presynaptic and postsynaptic. It's called synaptic because here's a synapse, okay? So the in here is a synapse, so presynaptic is before synapse, postsynaptic is after synapse. Also, they're presynaptic neurons, okay? It's just an adjective describing where the neurons are in terms of the synapse. Okay, so we left off here. So this would be the axon terminal here on this side of the presynaptic neuron, and this here would be the dendrites of the postsynaptic neuron. Remember, axon terminal is where information leaves. Dendrites is where information is received and passed on to the next neuron. So terminal, what do we do in airports when there's a terminal? We leave, right? So think of it as terminal, information is leaving, dendrites, information comes in. Okay, so moving on from here, the action potential is propagated along the axon all the way here, triggers a release of these little vesicles here. So these, you see the little things, and they're filled with little tiny sacs of little neurotransmitters here. So that is what we call a vesicle. And the action potential, when it reaches the terminal, it releases these vesicles. So we can see one here that's opened, and the neurotransmitters are being released, boop, 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 and they diffuse, okay? Keyword, diffuse. Diffuses across the synapse. Across the synapse. And then this reaches the receptors on the postsynaptic neuron. So the receptors, they take in these like little tiny neurotransmitters, and then It could either be excitatory or inhibitory, which tells them to pass on the information or stop that information, which we will move on to in just a couple minutes, or right now almost. So yeah, it diffuses across the postsynaptic neuron, reaches the receptors, taken in by the next neuron, and that neuron may or may not pass down that information onto the next neuron. So if it does pass it on, then action potential happens and propagated all along, and then this cycle repeats again and again, okay? So here we have neurotransmission in a nutshell. Okay, now let's move on to this. When we look at neurotransmitters, there are some main questions we can ask ourselves. So is it inhibitory, excitatory? These are just a couple examples I'm having here because they do fit the things I'm talking about right now. Are they agonist, antagonist, endogenous, or exogenous? Okay, they might sound very, very similar to you, but let's look at the prefixes here. So here we have inhibitory. Inhibitory. Okay, so inhibitory is like just like a negation, like unable. But anyways, it's for like negation. Excitatory, like excited. Okay, excited, I pass it on. Inhibitory, no. Inhibit, I don't pass it on. So this is pass on. No pass on. So say an inhibitory neurotransmitter were to be received by the postsynaptic neuron, it inhibits the transmission by either maybe occupying the space on that receptor and not allowing the neuron to take in the information released by the excitatory neurotransmitter, and therefore causes inhibition of the neuron. But excitatory neurons, when it attaches the postsynaptic neuron, it causes the neuron to be more likely to fire that signal onto the next neuron. So this one passes down signal, and it's like very, very like excitatory. Inhibitory, no, kind of stops the signal there, stops passing on, okay? So here we have acetylcholine and scopolamine. This one you will be learning in Antonova study. We do Antonova study in our personal like school, but you are free to do whatever study you feel comfortable with or whatever is taught to you. But here are two like neurotransmitters that we use to write our paragraphs. So of course acetylcholine would be excitatory, scopolamine would be inhibitory, because scopolamine is an inhibitor of acetylcholine. Okay, next we have to ask, is it agonist or antagonist? Now this is like some kind of fancy terminology which will appear in some questions, and it may catch some people off guard, because what do you mean agonist, antagonist? Because we're more familiar to inhibitory and excitatory, right? Well, it's practically the same thing. We mean the same thing, but there's just a tiny bit of difference in terminology. Agonist refers to excitatory, okay? Agonist is basically like causes an effect in the neuron, kind of like passes it on. It's active, makes the neuron more likely to fire. So agonist corresponds to excitatory here. But antagonist, you can think of as like a villain antagonist, you know what I mean? It inhibits a signal. So inhibitory. So again, antagonist, scopolamine, agonist, acetylcholine. Now, endogenous or exogenous. So endogenous means in. Exogenous means out. So is this neurotransmitter created in us, or is it naturally occurring in ourselves as humans, or is it exogenous, which means it comes from an outside source? Is it injected in you? Have you eaten it? Have you however by means did not produce this, but it has ended up in your body by like human measures? So basically just this is in your body, out of your body. Okay, so exogenous would be scopolamine and endogenous would be acetylcholine. Okay, acetylcholine is a hormone involved in learning and basically spatial coordination, all that kind of stuff, also concentration. So we would describe this as an endogenous agonist. This one would be an exogenous antagonist, okay? We like using this terminology. This terminology is very nice. Okay. Now let's move on to the possible SAQs. So they are pretty similar, some of them. You do have to pay attention to what they're exactly asking of you. So one neurotransmitter. So just one, okay? Just one. Very simple. You can choose scopolamine. You could, well, preferably you choose acetylcholine because, you know, it is very, very useful. And you have Antonova there. You could also choose cortisol, which you could use newcomer study for, but I don't think that fits here super well because that's more another topic. But please use Antonova, scopolamine here. One agonist. Okay, we talked about this. Scopolamine, easy. And then Antonova, and then one antagonist. Oh, sorry, this one. Acetylcholine is agonist. One antagonist is scopolamine. So acetylcholine ACH, and then ACH scopolamine. Okay, cool. I to know both of them. One study antagonist. Okay, this is just this question, but fancier. Okay, so just still scopolamine. Oh, sorry guys, I read the same thing here, so let me just fix that. Agonist. There. So again, same thing as ACH here, so just one agonist, talk about ACH, one antagonist, talk about scopolamine, of course, and then excitatory or inhibitory. This one you can choose either one, okay? So scopolamine or ACH, take your pick, take your choosing, whichever you feel more comfortable with, do that. Just since excitatory. So really pay attention to what the question is asking you here. It's actually quite simple. Just describe one. Don't describe both of them because you risk running out of time and you probably won't get marks for the other one because they did ask you just to describe one. So yeah. Okay. Let's look at some example paragraphs. So I'm aware these are not the best paragraphs. They are just kind of examples to show you what you should do. So let's explain neurotransmission. So of course, we talk about the whole process as a whole first. So the process, neurotransmitter, chemical, and then talk about the synapse, talk about the example of ACH. So what is it involved in? What does it do? Talk about the process of the electrical signal passing down the axon to the axon terminal, presynaptic neuron. And then action potential is the general name of this entire process. And then triggering the vesicles to open and release chemical messengers. And then the neurotransmitter, which I've abbreviated here because there's a lot of writing in this. And then, there's also exocytosis. Oh, I forgot to mention this. Okay, so exocytosis and endocytosis. So similar to exogenous and endogenous, it is exo is the leaving of a cell, like to leave a thing, to leave the presynaptic neuron, and then endocytosis is action of entering, so entering the postsynaptic neuron. So out, in, okay? So, and then they activate the receptor sites, produce an excitatory response, which is increasing the neuron's positive charge, making more likely to fire, or inhibitory charge, which is less likely to fire. And then it sets off the impulse again, and then on the axon to the next neuron. Okay, that's cool. And then we mentioned Antonova. Okay. This is our study paragraph. We will mention Antonova in a further video if you are interested in this. And then now we just conclude, how does this study show this, and how does this study show this, okay? So linking it together. So blah 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 conclusion, something just explain how the study shows the neurotransmission. Easy, okay? Now let's look at agonist or antagonist. So antagonists, I give a brief explanation. So drugs are substances which bind to receptor sites of neurons and block the binding of neurotransmitters, which we have talked about in this video. And then this reduces the effect of the neurotransmitter, and if this postsynaptic neuron does not receive the signal, it will not fire along to the next neuron. So this is kind of like a painkiller, okay? So say you take a painkiller, it's an inhibitory thing, it inhibits signal so you won't feel the pain anymore, which is your neurons less likely to fire, therefore why you don't feel enough pain. So, and then just talk about like how it was also useful in this study, and then our like sum paragraph. Okay, how does this study show this thing? Okay, and then agonist is amplifies an effective neurotransmitter by binding to the receptor and activating them, so they are ACHs. So ACH is an agonist to itself in a matter of saying. It is like an excitatory neuron, just as I said, works bit more different, but pretty much same thing. Yeah, and then okay. I kind of explained agonists a bit wrong in this video, I'm gonna be really honest with you, but this paragraph explains it better than I do right now. So agonists are very similar to excitatory neurons, except they do this in other ways. Excitatory neurons, they increase firing of a neuron by chemical means, but agonists, they block the reuptake channels of the neuron, and then this leaves like a great concentration of the excitatory neuron in the synapse. So let's run an example here. So presynaptic, postsynaptic, not the best drawing, whatever, we'll get over it. So this here, reuptake channels blocked. So when a neurotransmitter, on the presynaptic neuron, so the reuptake channels will be here. So these neurotransmitters, normally, if these weren't blocked, these would just get reabsorbed into the neurons, okay, and then stored it back into vesicles, and then the concentration of synapse here would go down. But this, when it blocks it, so it doesn't reuptake anymore, the neurotransmitters have nowhere to go except for being accepted into the neuron, postsynaptic neuron, okay? So it will have to go into the postsynaptic neuron. Therefore, the concentration being high really helps it going through. Therefore, an agonist is quite good for neurotransmitters. Okay. And here, as you can see, we just mentioned how I explained this, and then it's the study paragraph, Antonova again, and then how does this study paragraph show this, or how does this first paragraph show this? Okay, I think that's pretty simple, and that's all you kind of need to know for neurotransmission. I hope you enjoyed and learned a lot of things from this video. If you do have any questions, please leave comments, and see you guys next week. Thanks for watching.